Okay, hi everyone, welcome back, and thanks for joining us today for Neurolectives webinar. My name is Rafa Novak and I will be your host today. Today we'll be talking about transcranial alternating current stimulation from physiology to cognition. This is a really hot topic. We have about 800 registrations already and 200 attendees online. So I'm very happy to welcome our guest speaker. We are honored to have today Dr. Miles Wischnewski. Hi, Miles.
Hey.
Hi, good to have you. We'll begin with Miles' presentation, and this will be followed by Q&A period. So feel free to submit your questions at any time using Q&A tool. So please let me quickly introduce our speaker. Miles is a postdoctoral researcher at the University of Minnesota in the Department of Biomedical Engineering and part of the lab of Professor Alexander Opitz. He has a background in psychology and neuroscience, and during his Ph.D. at the Donders Institute in Netherlands, he investigated how decision-making and learning can be modulated through non-invasive brain stimulation. He quickly found that getting a good understanding of behavioral modulation through TACS is impossible to fully understand without neurophysiological markers. He specialized in EEG and MEG data before, during, and after TACS and other neuromodulation techniques. Then during his PhD, he spent 6 months at the Leibniz Institute in Germany in Professor Michael Nitsch's lab to specialize in neurophysiology and TACS. And at his current position at the University of Minnesota, he still investigates fundamental aspects of TACS. However, in addition, he is involved in a variety of clinical studies on both psychiatric and neurological disorders to investigate the potential therapeutic effect of TACS. So welcome, Miles. We are very happy to have you here. All yours.
All right. Thank you very much. Well, thank you for the kind words. It's always hard to take credit for all of that because, of course, there's always a team behind me and not behind me, also like supervisor and everything, that allow me to do all this stuff. But yeah, I'm super excited to be here and super excited to talk about TACS. The title is From Physiology to Cognition. And as Rafael already said, cognition is really where I come from. I want to understand cognition. I want to understand how humans behave, why they make certain decisions, how they learn, all that kind of stuff. And I always initially, from the beginning, thought that neuromodulation is a great tool to use to understand that because, if you change behavior through neuromodulation, you can understand which neural structures are important for that decision making. But then I also quickly found out that non-invasive neuromodulation, the effects of that are sometimes a little bit hard to understand because we don't always fully understand, at least in humans, what or how much of the actual neuromodulation actually reaches the brain, and also what effect that has. And without understanding that, it's really hard to make that transition to cognition. So I kind of want to go over what I learned over the last years, either through my own studies or things I've read from other people. Just a quick introduction. I think most people who join the webinar will have an understanding of what TACS is, transcranial alternating current stimulation. But the idea is that we apply an alternating current to the head in a non-invasive way. So it's not really painful. You might feel like a slight itch or something like that, but it's not invasive anyway. And then the idea is that some of that current that we apply to the head actually reaches the brain. Now, obviously, there is some intermediate tissue there. There's the skin, there's the skull, and there's the cerebrospinal fluid. So it means that not all of that electric current actually reaches the brain. But that there is a small amount that does actually reach the brain, and the idea is that that modulates neural activity. So again, most people will be familiar with TACS, but I kind of want to reiterate why I think this is so important and what's at stake here. Because TACS might be a great tool in the future for therapeutic application. And certainly in a time where, we get more and more attention for psychiatric disorders on top of already known neurological disorders, it's always great to have more tools that can be used to treat different disorders. And I think what is good about or cool about TACS is that it has a great safety profile. It is, the side effects are really minimal, except some tingling sensation, itching sensation, maybe a headache, but not much more than that. Compared to other things like TMS, fMRI, MRI, you name it, it's cheap. It's definitely affordable. It's portable, meaning that people eventually can take it home, and there are some clinical trials going on right now where people take home a neurostimulation device, either TDCS or TACS, and use it under guidance of a professional. And yeah, that brings me also to the next point. It's relatively easy to use. Now, obviously, I will discuss today that TACS isn't always super easy to use, and that to understand the effects, we have to really do some good research. But generally, applying the machine or applying the electrodes and then using the machine is not the hardest thing in the world. I'm sorry about that. And then one point that I really want to stress, and that will come back in the first part of my talk, is that TACS is really backed by neurophysiological evidence. We know it does something to the brain, and I will hopefully convince you of that as well. And that is important because even if it wouldn't do anything to the brain, there could still be some behavioral effects, right? There are methods where where we change behavior without directly affecting the brain. TACS could do this through tingling in the skin or the flickering lights that you might perceive. And maybe, it could theoretically be possible that all the behavioral effects we see are driven by that. But we do have some good neurophysiological evidence that it actually targets the brain and that we actually see some changes in the neurons. And I think that evidence is quite strong, particularly in recent years. There has been a growth of that evidence, and that evidence is getting stronger and stronger. And I would say it's probably the evidence for TACS having an effect on the brain is equal, well, maybe not equal, but very close to the evidence for neurophysiological effects of TMS. So I think there's some very solid evidence. And lastly, it is cool because it can be personalized. There are a lot of parameters that can be tweaked, and that is also great when considering that every person has a different brain, and every disorder is also different in different individuals. All right, so from biophysical principles, I don't want to talk too much about that. We use 2 or more electrodes to induce a current into the brain, and as I already talked about, as we can see here, not all of that current actually reaches the brain because most of it is blocked by the skull, and then even if it goes through the skull, there's the CSF layer that distributes the electricity a little bit. So it is always a little bit like TACS will never be as focal as, for example, TMS is. Even high density TACS, Ph.D. TACS, although it's more focal than maybe a 2 electrode setup, it will never be as focal as TMS is. But that doesn't necessarily need to be a bad thing. And then the overall idea, the base idea of TACS is that we induce this alternating current, which then doesn't cause action potential in the neurons directly, but it does bias when those neurons spike. And then the idea is that there's this phase locking to the local field potential, meaning that we get something which we call entrainment. So typically when we apply TACS, we apply it somewhere between 1 and 4 milliampere, although 4 milliampere is already quite high. That's not used so much. Typically it's around 2. And that translates to anything up to 1 millivolt per millimeter in the in the human brain. Although again, 1 millivolt per millimeter is quite high. So typically it's around 0.2, 0.3, maybe 0.5. And the deeper you go into the brain, the less the effects typically get. Now, I want to spend some time on explaining these neurophysiological effects of TACS, because in order to understand what TACS does to cognition, we have to first understand what it does to the brain. And again, I already introduced that there is this idea that TACS entrains neurons, not by inducing spiking directly, but by biasing the spike timing. However, that is only one of the mechanisms by that TACS induces, and it would be too simplified to just equate TACS to a machine that induces entrainment. And that's something I want to talk about a little bit. But first, let's talk about this entrainment and this idea that TACS changes the timing of spikes and aligns it to its own oscillation. We have various evidence from cellular slices in rodents and even in primates that this actually is the case. Now, the great thing is that consistent over these studies is that entrainment can be observed at intensities that we actually can use in human research. So, of course, you know, it could have been that TACS does, or that an alternating current does entrain neurons, but that it happens at an intensity that is way too high for human research, and that we would never be able to actually do it safely in humans. But that doesn't seem to be the case. It does seem to be the case that we actually can do this. And again, this comes from in vitro and in vivo research that spans for 50 years. So interestingly enough, is that there's a lot of research on alternating currents on neural activity that happened in the 1980s and even in the 1960s, which is long before the abbreviation TACS or the name transcranial alternating current stimulation was actually introduced. So there is, you know, a long history of evidence there. What we also see is that generally TACS is frequency dependent. And I will come back to that in 2 minutes, but that relationship isn't always super clear. Another point of contention that seems to be generally true, but not always, is that entrainment occurs mostly in pyramidal cells. But we always need to consider that within a network, there is always an interaction with inhibitory neurons, and those can be entrained as well. One other thing that we need to consider when looking at these animal studies is that a lot of animals are anesthetized, and anesthesia in itself has a big effect on brain activity. So when doing TACS research in anesthetized animals, we always have to consider that, you know, the actual dosing that we use there might be or needs to be higher, because an anesthetized brain just needs a higher stimulation. Yeah, so coming back to this idea of frequency dependency, this is a study, a computational modeling study, which promotes this idea of the Arnold tongue pattern. And the idea of the Arnold tongue pattern is that TACS generally has the strongest effects on neurons that have an intrinsic firing rate that is similar to the applied TACS frequency. So if you have neurons that prefer to fire at, let's say, 8 hertz, then the strongest effects will be if TACS is applied at around 8 hertz. And the stronger the TACS amplitude is, the more wiggle room you have. So even if you have a very high stimulation intensity, you might get away with stimulating at 9 hertz or at 10 hertz. But typically, the closer you are to the intrinsic frequency, the better your chances of entrainment. Now, you can also see that there are some harmonic effects. So when the frequency is twice as much, you will still see that some entrainment happens at oscillations that are twice as fast. So that's the idea that TACS is really frequency dependent. You have an intrinsic firing rate that is a certain number, and TACS should be around that number. However, that's not the entire story, because it does also depend on what type of neurons we're looking at. And this is a really cool study. It's a preprint on bioRxiv, and I'm sure it will be published very soon by Lee and colleagues, the group of Kostas Anastasiou, who have done a very cool study in a cellular slice where they apply electric stimulation, ES, which in their case wasn't alternating current stimulation, and they applied that to different neuronal types. And they had pyramidal neurons and two other kinds of neurons, which are inhibitory neurons. And the interesting thing is that when looking at the pyramidal neurons, they showed some significant entrainment. So on the y-axis, it's a spectral length, but in their case, it was basically the phase locking value, which is a representation of entrainment. They saw that at 8 Hertz, when stimulation was applied at 8 Hertz, neurons were entrained. And that makes sense because the pyramidal neurons they observed had an intrinsic firing rate of around 8 Hertz. But then when they used higher stimulations, there was still some significant entrainment, which kind of goes against this idea that it is super frequency dependent. Now, I'm not saying that it isn't, but definitely there can happen something at other frequencies as well. And already, as I mentioned in the previous slide, we typically don't see any entrainment in inhibitory neurons, but what they show is that that actually can happen, but at higher frequencies. At frequencies of around 140 Hertz, they observed significant entrainment in inhibitory neurons. Now, these kind of frequencies are not often used in TACS research. Sometimes they are, but not often. So that's why we maybe don't talk about that too much in human research, but it's definitely something to keep in mind. So yeah, we have this evidence in rodents, but obviously rodents are not a good representation of what happens in humans. But the good thing is that we, in recent 4 years or so, there is some more research in non-human primates. And although non-human primates are also not humans, they are the closest thing we have to humans where we can do invasive research. And this study by Johnson and colleagues really nicely shows this idea of entrainment, that in a baseline phase, which is here, this upper section, and by the way, the 3 different columns are different intensities of TACS, the spiking just happens at random intervals, whereas if TACS is applied, which is here in orange, you see that the timing of the spikes happens really in a certain window, and that window, in this case, coincided with the peak of the oscillation. And that's basically generally what entrainment is, that we bias the spike timing to a certain point in the local field potential, and the local field potential, we assume, is driven by the TACS. You can also see this in this so-called polar histogram. So the polar histogram is a representation of at what phase spikes happen, and in this case it happens around 0 degrees, which again is this peak phase. So this idea of entrainment is now also shown in non-human primates. You know, the idea of that TACS entrains neurons is really strong, but I said there is a little bit more than that. And this is a really cool study by Krause and colleagues, where they applied TACS at different intensities, again, in non-human primates, and they looked at which phase neurons were entrained. Now, I'm sorry, I forgot to put in the legend, but the blue color is when there is no TACS. So this is and again, this is a polar plot where the further you are outside from the middle, the more entrainment there is. It's called the phase locking value. So in the middle, there's no entrainment. On the outside, there is entrainment. So we see that even at baseline, when there's no TACS, neurons are entrained to their intrinsic local field potentials. And what they showed is that with TACS at a lower intensity, this intrinsic entrainment is kind of like destroyed. So there's kind of de-entrainment. But then when using a higher intensity in red, then the entrainment comes back. But interestingly, it happens at a different phase. So yes, TACS can entrain neurons, but it can also shift the the preferred phase of neural spiking. And that's interesting because we have seen something very similar in one of our own studies that is currently still in preparation, and we hope to be able to publish it soon and to put it in bioRxiv somewhere in the next 2-3 weeks, where we do see this gradual shift over time. So again, in the middle, there's no entrainment. On the outside, there is entrainment, and the different colors from light to dark represent time windows during stimulation. So we applied TACS and then looked over time, does it change? And we do see in some neurons, not in all, but in some neurons, this shift. And interestingly, all of the time, the neurons are entrained, but the preferred phase changes over time. And this is something which is seen outside of TACS research as well, and it's called phase precession, where for example, rats or mice that are put into a maze, if they walk through the maze and the spiking of hippocampal cells is recorded, their preferred phase tends to shift. And the idea there is that it is related to some learning effect or some synaptic plasticity effect. So what we see here is that TACS can do something that is really reminiscent of that. Which might suggest that TACS not only entrains the neurons, but by changing the phase also adds something else to it. And if we compare this to this phase precession literature, it might be something to do with synaptic plasticity. And I will come back to that in a second. One thing to note from all these research studies as well is that the firing rate is typically not increased or decreased. And that's interesting because that's quite different from what we see in other neuromodulation techniques like TMS or TDCS, where we do have the assumption that actually firing rate changes. With TACS, firing rate typically doesn't change, but it's the timing of when the spikes happen that changes. Another study that is really interesting that I unfortunately don't have enough time to go through is by Vieira et al. Showing that even if sensory and peripheral inputs are blocked, so if people, or in this case monkeys, don't feel or don't have any flickering sensation, these effects still hold. So it's not just a peripheral effect. And as I said, there is more than just entrainment, because the study by Johnson showed some burstiness, so that spikes tend to happen in bursts when they are at a certain phase, and also that the preferred firing of neurons, the phase of that seems to change with TACS. Now this then brings back also the idea of how much intensity do we actually need to stimulate to get this effect. And that's why my colleague Ivan Alexeichuk, he done a meta-analysis on all these studies that have been performed since the 1980s in rodents, in non-human primates, and in cellular slices, to see what is the minimum intensity that is needed to actually get entrainment or to change the neural spiking. And this is a graph that kind of represents a probability curve. So the probability, and we done this for brain slices and anesthetized rodents, which again, as you can see, you need a higher intensity to induce any spiking there. And then we also looked at awake and behaving mammals, which were either small rodents or non-human primates. And we did see that for them, you need less intensity to actually entrain neurons or to change their spiking behavior. And what you can see is this probability. So there's a probability of 50%. So 50% of all studies showed some entrainment that induced electric fields of 0.25 millivolt per millimeter. And again, this is a value that is easily achievable in human research. Again, 50% probability might not be that much. So if we, for example, look at 80% probability, we look at 0.3 to 0.4, which is still achievable in humans. So, with the intensities we use in humans, there is quite a significant probability that we can induce these neurophysiological effects. So I already hinted at that the changes we see in firing phase might have something to do with synaptic plasticity. And this is really important because the idea of entrainment, if you just take the idea of entrainment, that is something that would happen if TACS is applied and then basically disappears once TACS is turned off. And if we want to use TACS as a clinical tool, that's not great because we cannot have people walking around with a TACS device 24 hours a day. So it would be great if we could find some evidence for TACS having some after effects, some remaining effects, some plasticity effects. And this is a study, I want to discuss a study that I've done in Dortmund at the lab of Michael Nietzsche, where we kind of looked at, does TACS have after effects, and are these related to synaptic plasticity? So what we did is we applied TACS, but first of all, we had some basic physiological measurements, resting motor thresholds, some single pulse MEPs, and some paired pulse MEPs, motor evoked potentials, and a resting state EEG as well. Then, before actually applying the TACS, we gave people a drug intervention. And the drug intervention was either a placebo, so PLC, which has no effect, or DMO, which is dextromethorphan. And dextromethorphan is known to block NMDA receptors, meaning, that if synaptic plasticity occurs through NMDA receptors, which very often is the case, if these are blocked, then we would expect that any after effects that TACS has would disappear with this drug intervention. So just to make sure that the drug intervention itself didn't have any effect on these measurements, we did our baseline measurements again. Then we applied TACS for 15 minutes over the primary motor cortex, because again, we use motor evoked potentials as an output measure. And we used the beta frequency because that is a very dominant rhythm in the motor cortex. And then we looked at after effects for up to an hour, again, on the single pulse motor evoked potentials, paired pulse motor evoked potentials, and the resting state EEG. And again, the hypothesis is that we do see some after effects of TACS. Those would remain if we give the placebo, but they would be abolished with dextromethorphan in case this would be really a effect of synaptic plasticity. And fortunately, to make it really easy, that's what we found. So on the x-axis, you see the time. On the y-axis, you see the normalized motor evoked potentials. And again, the PLC placebo in this case stands for the drug intervention, not for a sham condition or anything. It's the drug intervention that is the placebo here. So with the placebo drug medication, we do see that TACS has this after effect spanning for at least 60 minutes. We should have tested longer if we would have known this. But then if dextromethorphan is applied, we do see that there is basically no after effect. And we do see exactly the same thing if we look at the beta power over the motor cortex in that same time frame. So this really suggests that indeed there are after effects, and these after effects are driven by NMDA-mediated synaptic plasticity, which is great. So besides the direct effect on spike timing and synaptic effects, there's also some evidence for effects on long-range connectivity. For now, I will not go into it too much because I have some slides later on. So the idea is that we can use a phase manipulation of TACS. So by using more than 2 electrodes, by using 3 or more electrodes, we can actually, rather than have 2 electrodes at an opposing phase, which is generally the case if you just have 2 electrodes, we can have 2 regions where the TACS current is in the same phase. And the idea of that is to synchronize regions that are really distant from each other, which might be really interesting because we know that there are some long-range networks in the brain. There's, of course, the resting state default mode network, but there are also working memory networks, frontoparietal, you name it. So synchronizing them would be great. And the idea is that if those phases are aligned, the spiking is aligned as well, and that's why we get synchronization. Whereas if that is not the case, if they're an anti-phase, so to speak, there's some desynchronization. All right, now I want to actually move this this idea of that we can have some physiological effects also to cognition. So can we actually modulate cognition with TACS? And I have here this painting because I always want to use this analogy that with TACS, we basically have all the tools. We know that it works on neurophysiology. We have the tools. So compare this to a painter. We have paint, we have a canvas, and we have ourselves and we have the ability to apply paint to the canvas. So all the prerequisites are there, but that doesn't mean that we're a good painter yet. And it's kind of the same thing with translation of TACS from physiology to cognition. We have all the tools. We know that it has an effect on the brain. That doesn't necessarily imply that we know what to do to affect cognition. And yeah, this is a figure from our very recent review in Trends in Cognitive Sciences, where we talk about 4 different aspects of physiology and cognition, how they are intertwined, and how TACS can be used to modulate both at the same time. The first example I want to give is about homeostatic cognitive systems. So what we do know is that cognition is complex, and cognition is not a single thing that you can just increase. You cannot just increase cognition. That's difficult because cognition often has opposing mechanisms. For example, there's a speed accuracy trade-off. There is the idea that there's stability versus flexibility and impulsivity versus sustained attention. So either you can have for example, some rewards, you can have them very quickly and satisfy that quickly, or you can sustain that, or you can have inhibit that and have a longer like a delay of gratification. And again, so these are just 5 examples. There are more trade-offs that happen in cognition, and the idea is that TACS it can push both of those dimensions sometimes a little bit. Like I think this plot is a little bit showing that you can improve both dimensions at the same time, but most of the time it will bias it towards either faster responses, more accurate responses, or more drive for reward or more drive for inhibition, something like that. And that is something to keep in mind. TACS is not a wonder tool that all of a sudden makes us superhumans and have unlimited cognitive abilities. Typically, if you increase one thing, it will be at the detriment of the other. And it's interesting because this is also related to physiological measurements in the brain. We know that there are oscillations in the brain that drive different things. For example, there's the theta-beta ratio, where theta oscillations in the prefrontal cortex basically reflect this immediate reward drive. So you want the reward and you want it now, whereas beta oscillations are more a cognitive control mechanism, which is more important for sustained performance. So if you want to delay a reward now for a bigger reward later. And the theta-beta ratio is well researched, and it has been linked to psychiatric disorders like ADHD, although again there are some discussions whether it is a good marker for that, but there's definitely some relationship there. So we did a study where we wanted to investigate this, and what we did is we gave people a reversal learning task, and the task is really easy. You have a condition where you can get points, either a high number of points or a low number of points. If you select a high number of points, you either get a lot of points, but if your answer is wrong, you lose a lot of points. So this is kind of a high risk condition because you either win a lot or you lose a lot. Whereas the low number is a low risk condition because you either gain just a little bit or you lose just a little bit. What is not known to people is that initially the chances of the high risk option be the best option is initially much better. So in 80% of cases, the high risk option is the best option. But then all of a sudden we switch that to the point where the low risk option is more beneficial. And this is kind of like a generic overview of how people would perform on a task like that. So initially they wouldn't know whether they want to do high risk or low risk. Then they learn that the high risk option is better. And then here, eventually there is a reversal. And then after a few trials, they understand, okay, maybe I should switch to the low risk option. And then eventually there's another reversal there. We wanted to see what theta TACS and beta TACS does. And what we found is that the theta TACS over the prefrontal cortex, it kind of moved this curve a little bit to the left, meaning that participants learned faster that they had to switch to the low risk option, or later on back to the high risk option. So interestingly, they learn faster. However, one thing that we also noticed is that as soon as they switched, they were also more variable. So although they switched earlier, they didn't stay, they were more impulsive. They were kind of like very alert that something like this can happen again. So they were constantly switching even when it wasn't warranted. So what I want to say here is that it had a positive effect in the sense that it made learning faster, but it had also that's in the negative effect because it made people more impulsive. Now with beta TACS, we saw something different. We did see that people didn't learn any faster. Their learning rate was equal to sham. But what we did observe is that when they had to take high risk, they actually always stayed at high risk. And when they had to take low risk, they always stayed at low risk. So their rule implementation, or their impulsivity was better. So they didn't want to switch all the time. They remained with what they learned for that point in time. So again, there wasn't a positive effect on learning, but in this case on rule implementation. And this totally makes sense with this trade-off between theta and beta oscillations in the prefrontal cortex, and the idea that theta is a driver for immediate rewards, whereas beta is a more cognitive control mechanism. Another process that happens in the brain quite a lot is our nested oscillations. There's a lot of evidence for nested oscillations, and the idea is that there's phase amplitude coupling between local and global processes. And typically local processes are fast oscillations and global processes are slower oscillations. And that is important for integration of information. Now that might sound a little bit fake, so I'll give you an example from working memory. And again, this is just an hypothesis, so this is not like solidly proven, but this is a cool hypothesis that has some evidence towards it. And I think it might be a little bit more complex than I will state it here, but it's still kind of cool. The idea is that there are nested oscillations of theta and gamma in the prefrontal and parietal cortex. And the idea is that the amount of gamma cycles within the theta oscillations is kind of a crude measure for how many items a person can hold within their working memory. So if you simplify it really easy, if there are 5 gamma oscillations within a theta cycle, then you can remember 5 items. Again, I don't think it's that easy. It's probably way more complex than that. But just for now, that's kind of a simple working model we have. Then the idea would be, if we either slow down the theta or fasten up the gamma oscillations, that might have a beneficial effect on working memory. And that's exactly what 2 studies found by Nina Wolinski and Akhturk et al. In the last few years, is that when theta TACS was applied to the prefrontal cortex during working memory task, the lower theta frequencies performed better, whereas the higher theta frequencies performed worse. And in the study by Akhturk here on the right, visual memory performance was better when TACS was applied. And this blue line is when the individual theta frequency was used -1 Hertz, whereas the yellow is the individual TACS was applied at the individual theta frequency. So let's say this was around 5, and the blue line was the frequency was around 4 Hertz. Again, I'm sorry that I forgot to put in the legend, but yeah. So this idea that slowing down theta might actually improve working memory. Then there's another study by my colleague Ivan Alexeichuk, who actually used cross-frequency TACS. So he used a TACS stimulation where he actually combined the 2 oscillations. And again, what he found is like he kept the theta oscillation stable at 6 Hertz, but then varied the the gamma cycles within that theta oscillation. And what he found is that basically compared to a control, the 40 hertz gamma on top of the 6 hertz theta didn't have much of an effect. But then if 80 hertz gamma was used, then there was this increase in effect size in working memory performance. And this remained also for 100 hertz and even for 130 hertz. And then obviously it drops down a little because then we get into a range that has nothing to do with gamma oscillations anymore, but is involved in other things. So again, evidence for this fact that that there is this interaction between fast and slow oscillations. Long-range coherence. Again, I talked about this before, where studies have tried to use an in-phase and an out-of-phase current to synchronize regions that are quite distant from each other. And the studies by Polania and Violante from quite a few years ago show that is indeed the case. So when TACS was applied at 180 hertz, which is the anti-phase condition, reaction time went up, so it means worse performance, and in phase it made performance better. And the same was observed in this study by Ines Violante a few years later. So showing that in-phase stimulation, at least if you look at distant networks, might be more effective than just your regular TACS. This is another study where they found something very similar by Helfrich and colleagues, but on performance on an ambiguous motion task. Again, I won't go into the details because it's just a summary of what they already found. And then this is a study by my colleague Ivan again. I forgot to put in the reference, but it's Ivan Alexeichuk, 2019 in Nature Communications. This idea of a traveling wave. So in these previous studies that I mentioned, they looked at TACS either in phase, so there's a 0 degree phase shift, or 180 degrees. There's the phases are opposite to each other. But what you can also do if you use multiple electrodes is to have different phase delays. And that kind of makes sense because the idea of in-phase stimulation is that two distant networks are active at the same time. But maybe they're not active at the same time, but the signal is traveling from, for example, the frontal cortex to the parietal cortex. If that is the case, that signal transmission takes time. Let's say that takes 10 milliseconds or so. Well, if it takes 10 milliseconds of a alpha oscillation, for example, is 10%, which means that there's a shift of 36 degrees. So maybe it makes sense to have these phase shifts that are not 0 or 180, but actually are somewhere in the middle. And then you get this idea that you can follow a traveling wave of information. So an information from, for example, the frontal to the parietal cortex or vice versa. In their paper, they showed that this is theoretically possible. There's still this is still ongoing research, so that I don't have too many actual results to show you. But I think this is really, really cool because it takes into account that signal transmission takes time into the brain, and that to me makes absolute sense. The last thing that I want to talk about relating to cognitive mechanisms is again a very recent idea that TACS might not just affect neurons directly, but also has an effect on the metabolic network. And there is this recent review by Marone Bixson, who suggests that TACS not only directly affects neurons, but it can have an effect on vascular activity. So the veins and arteries of the brain. And that is important because these arteries and veins, they transfer glucose and oxygen and glucose are important for neural firing. So maybe on top of the direct effects that we have on neurons, there might be an indirect effect through the vascular system. And that doesn't even take into account that there are other brain cells as well, glial cells, astrocytes, you name it, that might also be affected by TACS, and they influence neurons as well. So it's always good to keep in mind that TACS may have some indirect effects that we honestly today don't really understand yet, but that definitely, or like it's only logical that they would also have an effect on neural firing if they are affected by TACS. Again, this is really cutting edge research, really new, so there's not too many results yet, but I think this is definitely something we as a community should look into in the future. Now, one thing I want to talk about is the now that we know what we can do with TACS and how we can affect cognition, I want to talk about a few things to consider when applying TACS. So when using TACS, you have to make sure that you have a placebo condition, obviously a sham or an active sham, but that's typically not enough because we have more parameters that we work with. It would be great to have a location control so that we don't just stimulate, for example, the prefrontal cortex if we think that's important for working memory, but that we have a controlled site as well, another region that we think is not related to working memory. Now, this is all true for TMAS, TACS, TDCS, you name it, placebo and location. But then for TACS, there is another added factor, and that's frequency. And if we believe that frequencies matter, for example, the alpha oscillations are important for visual processing, theta oscillations are important for working memory, gamma oscillations are important for working memory, then if we solidly want to prove that that is the case, we also need a control condition where we have a frequency control. So, for example, you want to improve working memory with theta TACS, then you ideally would also have an active stimulation condition with, for example, alpha or delta TACS showing that that doesn't change it. Now, I understand that having all these three controls is very hard and might not always be feasible. I mean, look at my own studies. Most of them definitely don't have all three. So it's often not feasible, but it's definitely something to take into account and, you know, mention as a limitation. Then there is also other things that we can do to make our experiments better, and that is to do some electric field modeling, shown here on the right, which I didn't really touch upon up until now at all, but it is something we have seen in the last 10 years blow up. And that's the idea that we can use computational modeling to understand where the electric field goes with either TDCS or TACS or TMS. But then we also should consider that there are differences between induced electric fields. Brains look different. One person's brain looks different than that of another, and that also means that you know, we have to take that into account. Obviously, there are other things like brain state, peripheral side effects that we talked about before that need to be controlled for. Optimizing a task, if you have a task that is already where people already scored 95% accuracy on baseline, you're not going to see an improvement. So take into account floor and ceiling effects, and obviously having specific hypotheses. Now, I see that I'm running a little bit close to the end of my time, so I might skip a few things here, but so we already talked about peripheral side effects like tingling and itching sensation and phosphines, and those will obviously also have an effect. So it's important to control for it. But skin tingling, there are some creams that can be used to decrease the sensation, and for phosphines, you can use some kind of LED flickering to control for that. Another thing that I specifically want to mention, but don't want to go in detail about, is that is the thing of ActiSham, which is a placebo control that doesn't just ramp up and ramp down, which is the standard sham control, but actually uses electric field modeling to create a TACS montage or TDCS montage that induces as little current in the brain as possible. And it happens because electrodes are so close to each other that basically the electric current shunts over the skull without going into or the skin and the skull without actually reaching the brain. And this is really cool because you still have the same sensations through the entire experiment that you would have with an active condition, but there's just no current reaching the brain. So this is definitely something, if you haven't heard of that, check that out. And then also one more thing is that with electric field modeling, if we do that, it's always important to consider that when we do electric field modeling, we typically assume conductivities in the brain. So we think that the skull has a certain resistance, the CSF has a certain resistance, the scalp has a certain resistance, and we keep that constant. In reality, though, is that there is a big uncertainty there. So the conductivity of the scalp of one person is completely different than that from another person. So when doing electric field modeling, always take into account that the value that you get out of there. So for example, you do your E-field modeling and you get out 0.235 millivolt per millimeter. That's a very precise number. Just know that in reality, that precise number might not be as precise, and there's a confidence interval around that. I don't want to go into too much the clinical application. I just want to mention a few things. And that is when we want to use TACS in a clinical application, we have to consider whether it's the right method, because TACS is cool, it's a hot topic, but it might not be the best thing for all purposes. If you want to increase activity in a certain region, for example, by changing the firing rate, as we already discussed, TACS doesn't change the firing rate, it biases spike timing. So in those cases, TACS might not be the best option. And also it is important that we, up until now, talked a lot about increasing power and increasing synchronicity. That is not always the best thing in clinical application, because, for example, we know with Parkinson's disease, there is an over-synchronicity. The same is true for epilepsy. Synchronicity in the brain is too large, and there something like a closed loop approach or anti-phase stimulation can help to actually reduce synchronicity in the brain, which then helps those patients to reduce their symptoms. I'm going to skip over this, and I'm just going to go to the take-home messages, which is that I think that it by now is quite clear that TACS has some solid neurophysiological effects, and that it is more than just entrainment. When taking into account cognition, we have to consider that cognition is complex. It's not just a one-dimensional thing, but it has multiple dimensions. In the figures that I showed on the right, it was 2 dimensions, but often there are 3, 4, 5 dimensions. So using TACS to modulate that is not trivial. And TACS will probably not improve all facets of cognition, but just a few variables. And when doing your investigation, it's important to have specific hypotheses on the variables you think that TACS actually is going to change. And performance follows an invert U shape. Again, coming back to what I said just a moment, improving synchronicity and improving power might be good in a lot of instances, but not always. Sometimes you want to do the opposite. And then for clinical therapy, there's still a lot of unknowns. That's basically the one thing I should say, because we don't know yet what multiple sessions do. We don't know what TACS applied for 6 weeks does. So these are all things that we still need to do research on. All right, with that, I want to thank you. I also want to cheaply plug my own YouTube channel right in the corner there. Yeah, and with that, I thank you for your attention. I'm sorry that I went a little bit over time, and I'll take questions.
Okay, thank you very much, Miles. So we open the Q&A slot. If you have any questions, we'll have time to take a few questions to Miles. So before we get the question from audience, actually, I would like to have take this opportunity to ask you what is your opinion regarding the entrainment using TACS in comparison with sensory stimulation, and this is in the context of the recent paper by Professor Buzaki group, showing that actually 40 Hertz sensory stimulation with the light does not entrain gamma oscillations and actually is producing the steady state response, and this was in Alzheimer's disease model. How do you compare this with the TACS?
Yeah, that's a great question, because with TACS, what we sometimes have as a sensory side effect is phosphenes. Particularly if you use stimulation at 10 hertz, and if you are more frontal, then you will have some flickering sensation going on. And if those flickering would directly entrain neurons, then we wouldn't know whether TACS is actually affecting the brain or whether it's the flickering that you observe. And I think the recent paper by Wischnewski and his group is actually good news for TACS, because I know there's an entire research line, wanting to use these kind of flickering sensory stimulation to entrain the brain. And then, for them, it's bad news that they kind of showed that it didn't work. But for us as a TACS community, it's good, because it seems to show that the phosphines aren't a huge problem. Now, I should caveat that by saying that there are other studies than just that group, from the Wischnewski group, and there have been some other studies suggesting that phosphines might affect TACS effects as well. Although we always should consider that those effects would be in the visual cortex, right? And whether those then translate to other regions in the brain, that's hard to say. So yeah, generally, I think that that paper is really good news for us, but it's still important to consider and minimize phosphines with TACS.
Okay. Thank you very much. So we have some questions. Let's take the question from Dustin, actually, and this was related to the reversal learning paradigm. The question is if you could dynamically switch between theta and beta stimulations based on the phase of the reversal learning paradigm. So you are in the increased both learning speed and rule implementation, like beta once it simultaneously. So actually you've showed this was possible in the next slides, actually, but yeah, go on, sorry.
Yeah, that's a cool setup for a study, and it's definitely something I would want to try. We haven't done it directly like that, but yeah, that's kind of that is cool, and I think that is also showing something that I really didn't mention too much, but this idea of closing the loop, maybe not necessarily closing the loop, but also individualizing and personalizing TACS on what's going on in the brain right now. So if you know that in some stage rule implementation is the most important thing in a task, then you try to boost that, and when there's a reversal, you go to the theta stimulation to boost that. Yeah, I think that's really cool. Of course, when you use a task, you as an experimenter when the reversal happens. The participants don't know, but now in other tasks, or when you just want to go by physiology, that might be a little bit more tricky. So for example, we see a drop in beta activity, and that's when we want to boost beta activity with TACS. Well, that would only work if we could use TACS and EEG or MEG at the same time. But we also know that that combination is very, very tricky, particularly with EEG. It's basically almost impossible. There are some studies suggesting cleaning methods, but it will always be hard to read out EEG and do TACS at the same time. So if you can predict a situation with this dynamic changing of frequencies in TACS, is really cool. If you can't predict a situation, you don't really know what you're doing. But yeah, it's definitely a cool setup. Yeah.
Okay, thank you very much. A very basic question at the beginning from Kairana Tsapkini regarding the entrainment mechanism. So there's an example of the patients with abnormality in specific frequency, for example, delta band. And what will be the recommended application of TACS? It means you will stimulate with delta or some other frequencies.
Yeah, that will, if we were to just know that, it depends on the abnormality, and it would depend on the abnormality. I have to go back to this example of epilepsy because there has been there are some ongoing clinical trials there that have not yet been published, but they will be published soon. Where epilepsy is, of course, is high frequency over synchronization, and there they show that the lower frequencies or desynchronization of that using an antiphase paradigm is really effective. So when there's an abnormality in the delta band, for example, if it's too, for example, too synchronized, I don't know too much about the delta band, then you would want to think about some kind of antiphase stimulation. However, if, for example, delta power is too big for some reason, then you might want to switch to another frequency, and preferably another frequency that is in balance with delta. So, for example, with delta, I know that there are also some ratios between delta beta and delta alpha. So, for example, increasing alpha activity will then will also change the delta activity. So it really depends on the abnormality. And yeah, I think generally in patients, a lot more research needs to be done before we really understand what happened in the abnormal brain, because everything I talked about today is what's going on in the normal brain, not even talking about the abnormal brain. So yeah.
We'll move one day to the clinical applications and the abnormal brain, actually. And there is a question from Judith: how do you target deep tissues specifically? How do you get into deep regions of the brain with TACS?
Yeah, with TACS, as it is known now, I would say it's not possible. However, having said that, there's of course this very recent technique, temporal interference stimulation, which in sense is TACS. I mean, it's still an alternating current, where the idea is that you have two alternating currents at an extremely high frequency in the kilohertz range, but there's a small offset there. For example, one is 2000 hertz and the other is 2005 hertz. So the difference is 5 hertz. And the idea is that because they're so extremely high in frequency, that they penetrate, or that they penetrate into the brain and don't really have a strong effect on the neurons, because neurons don't tend to operate at these high frequencies. But then where the currents come together, that's where you can get this 5 hertz amplitude modulation, because that's where the current combines, and that's then a way to get deeper into the brain. Now, I should say that there is quite a debate on temporal interference stimulation. There have been modeling studies suggesting that, for example, the high the kilohertz range of TACS does actually do something to the intermediate neurons. So the idea that kilohertz frequency doesn't affect intermediate, like the neurons, others like may contend that, and then it's really hard to distinguish where your effect is coming from. Is it coming from the 5 hertz in the middle, or is it coming from the kilohertz range from on the outside? So there's ongoing research, but no solid answer yet. But I think that's our best bet for now, to go with deeper tissue, at least if you want to use oscillations. Otherwise, if you want to go deeper in another way, of course, this ultrasound, focused ultrasound stimulation, which is now coming up, but I'm really not an expert on that, so I'm not going to comment on that too much.
Perfect, thank you. There is a question from Juan. Actually, you were not touching the topic of EEG recording during stimulation, but there is a request if you have an update of the status of the art of clean up EEG signal with simultaneous TACS.
I don't. So there are some papers from 2014, 2016 by a group of Christian Herrmann and some other groups that have used PCA analyses to get rid of the stimulation artifact. And then there's other ways, again, where you have these high frequency TACS and then this amplitude modulation, which might be easier to clean. But since these papers have been out, I am at least not aware of any other papers that have come up with better methods. So, and these methods are like a very good approach. It's just that it's very hard to distinguish what is actually, or how much of the artifact is actually remaining. Because even if it's just 2% of the artifact remaining, you clean 98% of the artifact, still that 2% might explain differences you observe in your EEG. So that's kind of the tricky thing with combined TACS and EEG. And I'm not aware of any methods that are able to get 100% rid of the artifact, at least not with EEG. There might be some, if you do intracranial recordings, intracranial EEG, there it might be possible, but not in standard EEG.
Okay, perfect. Thank you, Miles. We are running out of time. We have about more than 20 questions still. So I will, if you give a permission, I will forward you these questions from all audience to answer. And so thanks much for the presentations. Thanks for all questions. And I want to thank everybody for joining us today. And I hope you will join us again for the next webinars. We'll have Professor Carsten Wolters from Münster, Germany, and he will be talking about individualized targeting and optimization of multi-channel TDCS in focal epilepsy. So please visit our webpage and register for the next webinar. On our webpage, neuroelectrics.com, you will find more information. So thank you again. Thank you, Miles, for being with us. It was really a pleasure.
Yeah, thanks for having me.
Thank you very much. So have a great day and see you next time. Take care. Goodbye.
Thank you, everyone. Bye-bye.
Thank you.